It's for me a great pleasure to introduce you someone that probably most of you, if not all of you, know very well, which is Dr. José Luis Lanciego, which is much better known as Lancy. Lancy is a medical doctor from the University of Salamanca, also PhD in neuroscience from the same university. Later on, he moved to Amsterdam to the Free University as a postdoctoral fellow under the supervision of Dr. Wouterlot. I'm not sure whether my Dutch is good enough to pronounce these names, but after returning from Amsterdam, he was recruited by the University of Navarra to further lead the Laboratory of Functional Basal Ganglia Neuroanatomy, where he's still working. His research interests include a number of projects dealing with the pathophysiology of Parkinson's disease, mostly using non-human primate animal models. Among other boards, he's being a board member of the International Brain Research Organization, IBRO, and he's actually chair of IBRO until the end of 2025. He's also a board member of the European Brain Council and the International Basal Ganglia Society Council. He has co-founded Handle Therapeutics, a biotech company, pushing forward the development of novel gene therapies for Parkinson's disease. And today he will talk about, as you see on your screen, development and characterization of a non-human primate model of disseminated synucleinopathy. Sorry. So, Lancy, thank you very much for being here and for your willingness to share with us your work. And the floor is yours. So please go ahead with the presentation. Thank you.
Okay, thank you, Miguel. Thank you very much. Thanks to the organizers for giving me the opportunity to be here today speaking about things that are ongoing in the lab. Indeed, this is a great pleasure to see so many friends connected online. I hope to have a chance to meet all of you in person at the next CyberNet meeting to be held in Bilbao by mid-April. And by the way, Bilbao is a wonderful venue. It has a situation that is equidistant for any other place. Bear in mind that, as you may know, Bilbao is located right in the center of the universe. And indeed, Bilbao has a great beach named La Concha that belongs to the so-called Gran Bilbao. It's an area with geographical boundaries that are pending to be properly defined, comprising not only neighboring cities such as Castro Urdiales and San Sebastian, since it can probably be extended up to Algeciras. But well, once I have provided you with the right geographical context for the next CyberNet annual meeting, it's time to go a little bit more formal, getting started with today's webinar. As Miguel has mentioned, it's entitled Development and Characterization of a Non-Human Primate Model of Disseminated Synucleinopathy. Okay, so this is just a landscape view of ongoing projects in the lab. As you can see, we are using gene therapy tools with a dual purpose. On one hand, we are using AAVs for the purpose of disease modeling. And we have two main programs ongoing. One is related to overexpression of human tyrosinase in collaboration with Miquel Vila in attempt to set up a model of pigmented non-human primates. And the other one is related to overexpressing the alpha-synuclein, the mutated alpha-synuclein gene, A53T mutation, to set up a model of disseminated synucleinopathy. On the other hand, we have a number of programs that are more therapeutically oriented. And we have 4 programs going in parallel. One is related to glucocerebrosidase, another one with Parkin, and 2 more that, so far need to be kept undisclosed. For the purpose of today's talk, we are going to be more narrowly focused on this part of the activity here. So I just want to draw your attention to, this was a symposium held quite some time ago in 2010 in Germany. It's called the Herrenhausen Symposium. Probably is not the right pronunciation. So at that time, there was an initiative of Juan Carlos Lopez, the former, at that time, Juan Carlos was the former chief editor of Natural Medicine. So Juan Carlos, he managed to recruit more than 100 big names in the field of neurodegenerative diseases. They joined together for a couple of days, and they were challenged with the question of, okay, we know quite a lot about the neuropathological hallmarks of neurodegenerative disorders. We have gained an impressive amount of knowledge related to the mechanisms, underlying mechanisms of these disorders. However, the fact is that we are still lacking in disease-modifying therapeutics, and well, they were asked to identify a number of bottlenecks in an attempt to go, to have more successful clinical trials. So after a couple of days in Germany, and probably under the influx of a high serum level of ethanol, they decided in the end to come up with a list of 5 bottlenecks. And which is these 5 bottlenecks, the one that they stressed really very much, is that they agree that the main reason for failure, for repetitive failures in clinical trials, is that we have to recognize that we don't have animal models that are good enough. It is expected that, having better animal models, with this type of animal models, we can get more insight about which is the biology of the disease. We can find better biomarkers, and of course, we can design better preclinical trials with the ultimate goal of, increasing the chances of success of future clinical trials. Okay, so that's, an overall view of the problems we face when coming to develop new drugs for CNS disorders, particularly neurodegenerative disorders. So for most of the animal models we use, even in the best case scenario, these models only reproduce a minimal fraction of what is the real disorder. So these disorders are multifactorial. They have most often mixed pathologies. So not surprisingly, everything we can develop in in biased animal model, I would say, are hardly translatable to the real disorder. And that's the problem, and we need to find solutions for this bottleneck. And that's the main concern. So when dealing with dementia with Lewy bodies and Parkinson's disease with dementia, these two disorders are characterized by the presence of a widespread synucleinopathy all over the cerebral cortex and simultaneously in the substantia nigra. This is the typical, let's say, neuropathological signature that is common to both disorders. So to what extent, Parkinson's disease with dementia and dementia with Lewy bodies, to what extent are the same disorder or different clinical entities is an ongoing debate. It has yet to be solved. Depends, mainly to which is the person you are talking to. If you just ask a clinician, a neurologist, it is very likely that a neurologist will say that these are different disorders with different clinical presentations. However, if you talk to a neuropathologist, most neuropathologists will say that Parkinson's disease with dementia, and dementia with Lewy bodies are likely the same disorder, but in different stages of progression. So, well, that's something that need to be taken into consideration. So I have taken advantage of this webinar to introduce you a new ongoing debate. These were a couple of papers published last week in Lancet Neurology, where there are a number of, let's say, neuroinfluencers that are pushing forward the setting up a novel staging of Parkinson's disease and related synucleinopathies, but are supposed to be, this classification is supposed to be biologically based. They are taking advantage of the so-called alpha-synuclein seed amplification assay. So they propose, they postulate that we need to change our understanding. We need to forget about Parkinson's disease with dementia, and dementia with Lewy bodies. And all these disorders together are going to be renamed as neuronal alpha-synuclein disease. So the debate is there. From the two original papers, you can see that in the same issue, there are a number of accompanying comments with some people in support of these new ideas and some people against this way of, this way of rethinking Parkinson's disease and related synucleinopathies. It's not that much related to the talk today, but just keep an eye on this, because I am quite pretty sure that the debate has just started, and we will see more and more things to come into this focus in the forthcoming months. Okay, so coming back to the models, we need animal models reproducing later stages of Parkinson's disease and dementia with Lewy bodies. This is quite an unmet need. And when thinking about a model, the first thing you need to keep in mind is what exactly do you want to model? You want to get, in the best case scenario, as close as possible to the neuropathological signatures that typically characterize the disorder you want to model. And when dealing with these diseases, both diseases share a widespread synucleinopathy all over the cerebral cortex, together with alpha-synuclein expression in the substantia nigra. So this is what you want to reproduce. Then next step is to think about which is the best strategy to fulfill these requirements. So here we are taking advantage of a viral vector. It's an adeno-associated viral vector, serotype 9. So serotype 9 is an important choice, because we know that AAV9, at least in non-human primate brain, is capable of spreading very efficiently in the retrograde direction. So once you deliver AAV9 into the putamen, it is expected that every single neuron in any part of the brain that is innervating, is connected to the area of delivery, are going to be, the vector, the viral vector, will spread in the retrograde direction and transduce all the neurons, all the parent neurons that are giving rise to this type of projections. So in brief, by one single deposit of a given AAV9, you can induce a widespread expression of the encoded transgene by retrograde spread. So in brief, AAV9, at least in non-human primate brain, behaves the same way as might be expected for a traditional retrograde neuroanatomical trace. Okay, so in the end, this to properly characterize this model, in the end, this is going to be a simple, quite straightforward biodistribution study. And biodistribution studies are typically being made of three different readouts. You need to quantify the number of neurons, in our case, the number of neurons that are transduced with alpha-synuclein. Once you quantify the total number of neurons, another piece of evidence, another readout, is which is the rostrocaudal distribution of these neurons within a given area, within a given region of interest. And finally, another important readout is to elucidate which is the accurate location of these neurons. So any biodistribution study, need to take into consideration these three different levels of readouts. So in order to quantify things, and you have to keep in mind that, a non-human primate brain or a non-human primate section of the brain is quite a large piece of tissue. So you don't want to spend, hours or weeks behind the microscope just quantifying things. So some years ago, probably it was by late 2018, we decided to go for a license of something called IFORIA. IFORIA is the acronym of Artificial Intelligence for Image Analysis. has been developed by neuroscientists working together with engineers at the University of Helsinki. And this is the original paper of introducing aphoria by the group of Mark Sharma and Mikko Airawara. And when compared to traditional stereology, they realized in this paper that aphoria has up to 96% of accuracy when compared to traditional ways of counting things in the brain. So in brief, aphoria is cloud-based, so you don't need to install any piece of software in your computer. So you just simply upload the images to the cloud. You need to annotate the things you want to recognize in your samples, and to create an algorithm that can be later on trained to recognize the things you want to recognize. Once the algorithm is properly designed, it can be validated and released. And once the algorithm is released, counting things is fully automatic. You even have an option, just in case you have more money than time, to delegate counting and to delegate the setting up the algorithm to the company. I don't know about price, but just in case you have more money than time, everything can be done by the aphoria staff. So in order to show you how things operate, here we have a couple of sections for the non-human primate cerebral cortex. This is the, if I remember correctly, this is the supramarginal gyrus in the left hemisphere, and this is the type of counting you can achieve with the algorithm. So I have a small video clip here just to show you how things operate. So what you have in the left are different projects. For all these projects, we choose one of these two animals of the study. You can see this project is made of 110 sections. These are coronal sections of the monkey brain, that are serial sections. So it's just a matter of choosing any given section. So for instance, you have a section here where there are a number of things you can easily recognize. So for instance, this is the posterior cingulate, postcentral gyrus, superior parietal lobule, supramarginal gyrus, temporal lobe, the hippocampus, substantia nigra, thalamus, and so on. And then you have to quantify things. You want to quantify the number of neurons that are expressing alpha-synuclein. You can see there are quite a few. So the way in which things operate is quite straightforward. First thing you have to do is to draw the limits of the region of interest. So for demonstration purposes, here we have chosen the supramarginal gyrus in the left hemisphere. So you just put it in here for your records, supramarginal gyrus left. And then you have to choose the algorithm, because we have quite a long list of algorithms. We have chosen here an algorithm designed for the detection of alpha-synuclein neurons. It's something that you can share with others by email, or you can keep private for yourself. And at some point, as you can see here, the algorithm starts to operate. So in a matter of a couple of seconds, you have all the quantification being made. So of course, you can go to higher magnification to be sure to what extent the algorithm is accurate enough. You have neurons with different levels of expression of alpha-synuclein, and you need to make sure that the algorithm you have created is able to capture all different intensities of neurons expressing a given level of alpha-synuclein. So you can go to higher magnification just to make sure that everything has been counted, and there's not a single neuron be left uncounted. So here, this is made of 478 neurons. You can export the results to Excel. You can share with everybody by email. And this is, a quite straightforward way of counting things in a matter of a few seconds per region of interest, and saves an incredible amount of time. And indeed, another advantage is that the algorithm is making the counting for you. I mean, compared to traditional stereology, there is no bias in terms of the observer. So for instance, when we quantify neurons in the substantia nigra, it takes about 1 hour and a half per section for an experienced users of stereology. In the ideal scenario, you need to, to average the countings of two different users to get rid of this observer bias. This is something that you don't have to be of concern when using Euphoria. So if the algorithm is prepared good enough, you can rely on the algorithm for counting purposes, and it's an incredible amount of time. Okay, so let's move forward. So this is what happens at the level of the injection site. So these animals receive up to 3 deposits of the AAV9 coding for mutated synuclein into 3 different locations of the putamen. So what you have on top are several ventriculographies. This is the way in which we used to perform stereotaxis surgery in non-human primates in the old scenario. Now we have a new navigator. So in brief, compared to rodents, we cannot rely on cranial landmarks for stereotaxis. So we first deliver with a needle that is inserted in the lateral ventricle, in the frontal part of the lateral ventricle. We deliver an X-ray contrast that allow us to delineate the entire ventricular system. So this is the frontal lateral ventricles going parietal, occipital, and temporal. This oval area here is the thalamus. It's surrounded by the third ventricle. This is the aqueduct going all the way down to the spinal cord. So with this type of X-ray ventriculography, we can allocate the position of the anterior and the posterior commissure. We can delineate the so-called ACPC line, ACPC plane, that is our stereotaxic space of reference. And then with the second needle, this is the needle we use as a Hamilton syringe. This is the needle we use for delivery as a delivery device. So you can see, I hope you can see here, that the first injection has been done at the level of the anterior commissure. One more deposit is 1 millimeter rostral to the anterior commissure, and the final one is 1 millimeter caudal to the anterior commissure. This is the way we perform stereotaxis surgery in non-human primates. So then we stain for alpha-synuclein. The first thing we need to calculate is the area of transduction. So here the goal was to calculate how much of the putamen volume has been transduced with these 3 injections of the AAV suspension. So here this is a coronal section taken at the level of the caudate and the putamen. You can see the internal capsule going out here. This is the anterior commissure, and this is the second animal, caudate, internal capsule, anterior commissure, and the putamen. You can see that a large area of the putamen has been transduced with the AAV. We can calculate which is the volume of the putamen and the volume of the transduction. This is illustrated here. On average, in both animals, we have transduced roughly about 70% of the total volume of the putamen. That is quite a pretty good result. And when going from rostral levels to caudal levels of the putamen, here the area below the curve in gray illustrates the total extent of the putamen compared to the total extent, the total rostral caudal extent of the area transduced with the AAV deliveries. Okay, so with this type of material, then we can start counting things. So the first readout, as I mentioned before, is total number of neurons. How many neurons do you have in a given region of interest? These are the 2 animals of the study. You can see, for instance, the prefrontal gyrus, the superior frontal gyrus, and the anterior cingulate are the areas with the highest number of neurons. You have a nice representation of the insular gyrus on the left side, substantia nigra, and the thalamus, and so on, a few other areas with a smaller number of neurons. You can see both animals are roughly the same. And we also have a number of neurons in the contralateral cortex, but we have always the trend to neglect that corticostriatal projections arise from the left and the right hemisphere. Of course, it's not at the same level in terms of number of neurons, but you can see the superior frontal gyrus and the prefrontal gyrus in both animals are the areas in the contralateral hemisphere showing the highest number of neurons. This is not surprising because most of the areas that are, I would say, motor related, such as the superior supplementary motor area, primary motor cortex, and so on, They are all located in both the superior frontal gyrus and the prefrontal gyrus. Okay, so this is the first readout, number of neurons. A second important piece of evidence, a second readout, is the rostrocaudal distribution. Is by going this way, what we want to calculate is because it's not the same, I will say, to have, let's say, 1,000 neurons in a given region of interest, which is going to be the distribution of a given 1,000 neurons in the cortex. Is homogeneous or it's not homogeneous. So here we have a number of cortical region of interest, number of subcortical regions, and the extent, for instance, here, the extent of the superior temporal gyrus is calculated here, going for 4 millimeters rostral to the anterior commissure, back to 14 millimeters caudal to the anterior commissure. So the total rostrocaudal extent of the superior frontal gyrus in this animal is made of 18 millimeters. So we can count, we can illustrate the intensity of this labeling to illustrate to what extent the obtained neurons expressed as alpha-synuclein are covering the whole rostrocaudal extent of a given region of interest, or to what extent they are concentrated in a given area. So this is a good example is the postcentral gyrus. You can see the postcentral gyrus is made of 9 millimeters in terms of rostrocaudal axis, but the highest number of neurons were all concentrated in the caudal, in the rostral half of this postcentral gyrus, and not that many neurons being located in the caudal half of the postcentral gyrus. Okay. There are a few more useful information here. For instance, the insular gyrus has a nice number of neurons in the left hemisphere, but not a single neuron in the contralateral hemisphere, meaning that the projection from the insular gyrus to the putamen is strictly unilateral. So what else? The third readout that is also important is location of neurons. So for instance, when looking for neurons in the cerebral cortex, of course, it's not the same to evaluate whether or not these neurons were equally distributed throughout different cortical layers, or to what extent distribution of neurons is layer specific. So for instance, here we have some maps illustrating rostral, middle, and caudal levels of the non-human primate brain. This is an example of 4 millimeter rostral to the anterior commissure at the level of the anterior commissure, and 9 millimeters caudal to the anterior commissure of both animals. This is the injection sites. And you can see a stripe of neurons. I hope you can see this is, it's a very small magnification. These stripes were made of dots. A dot is just a single neuron. So you can see there is a layer-specific distribution of neurons expressing alpha-synuclein. These neurons are all located in layer 5 of the cerebral cortex. Here are some photomicrographs. For instance, this is the anterior cingulate gyrus, and you can see this a stripe of neurons all located in very deep layers of the cerebral cortex. This is the contralateral representation of neurons. Superior frontal gyrus, left and right. Prefrontal gyrus, that is comprised a very high number of neurons. The supramarginal gyrus, and the posterior gyrus, postcentral gyrus, that sometimes transduction is big enough to see neurons in Golgi-like labeling. This is a layer 5 pyramidal neuron with some basal dendrites and an apical dendrites typical for pyramidal neurons at that level. And this is the substantia nigra of the same animal. So this is a coronal section of the non-human primate brain. This is the midline. This is the red nucleus here. And this is the substantia nigra, where we have counter-stained with an initial stain. Here, high magnification, you can see that, for the neurons that are in the substantia nigra, about 50% of these neurons became transduced and started expressing alpha-synuclein. The main advantage is that since we know that these neurons express synuclein because of the retrograde express of the EEV, we know which is the identity of the neurons, so we can be sure that every single neuron expressing alpha-synuclein indeed is a neuron that is innervating the putamen. So at the level of the substantia nigra and at the level of the striatum, so here we have a stain for tyrosine hydroxylase. This is a coronal section of the non-human primate brain. This is the midline, lateral ventricles, fornix here, anterior commissure. This is the caudate and the putamen on the right side, the caudate and the putamen of the left hemisphere. You can even see one of the injection sites, where we quantify the optical density of TH positive terminals here and here compared to the other side. You can see that there is no difference in terms of the number of the density of dopaminergic terminals going from the nigra to the striatum. If you go far more caudal, you have a midline. This is the substantia nigra. This is the red nucleus here and here. This is the caudate, the tail of the caudate, dorsal and ventral. This is the caudal putamen, caudate and caudate, and the caudal putamen. And you can see even to the naked eye, it's hard to see whether there's any difference in terms of the number of TH positive neurons. So once quantified with aphoria and compared to the contralateral side on both animals, you can see there is no difference left and right in these animals, neither into this other animal. So the message here is that since these two animals, although they have a nice alpha-synuclein expression in the substantia nigra, these animals were sacrificed with a very short follow-up of just 1 month post-injection of the AAVs. So we are quite sure that 1 month of transduction with alpha-synuclein might not be at a time range long enough to induce any type of degeneration of dopaminergic cells. Otherwise, this degeneration will be quite clear with a TH stain. So we need to wait a little bit longer, and that's for sure. So nevertheless, by going this way, we can induce what we were looking for. So these animals have a clear and disseminated synucleinopathy throughout the cerebral cortex, from very rostral parts of the cerebral cortex up to the visual cortex in the posterior side of the brain. And at the same time, these animals also exhibit a nice synucleinopathy in the substantia nigra. That's, we have just submitted the paper to, and it's available in a public repository, as indicated there. So this is my team. They are the ones that are doing the nice job. I'm just nothing but the bureaucrat of the team. And for sure, I also want to acknowledge the funding agencies that are covering this type of work. So that's all for the time being. Now, for sure, more than happy to take any questions you may have in mind. Just to stress the fact that, just in case there's any of you that wants to use AAVs this type or any other type of AAVs we have in the lab, you need to be sure that these AAVs are just tools we have created, but are at your full availability. So if there is anything you need in terms of viral vectors, feel free to drop me a message, and we will be more than pleased in providing you with any AAV you may need it. So thanks a lot for your attention, and now let's go to the debate, just in case there are any questions.
All right, thank you very much, Lancy, for a very nice presentation. So the paper is now open for discussion. As I said earlier, if anyone is interested in posting a questions, please do so by raising hand using the raise hand option in your screen. Or if you don't have a microphone available, please send a message through the chat, and I will be happy to transfer the questions to Lancy. Okay, so while we wait for the first question, let's remind everyone that this webinar is being recorded, and the recording will be available at the CIBERNet website in a matter of a few days, a couple of weeks maximum. And also reminding everyone that the CIBERNet webinar series are held every other week on Thursdays at 3:30 p.m. And if you're interested in double checking the schedule for the next few months, go to the CIBERNet website, and I will have the full program till summer already posted there. Okay, so any questions? I don't see any raised hand yet.
Very easy job. Thank you.
Let me check the chat. No questions coming through the chat either.
Oh, fantastic. Yeah. Control.
Okay, don't be shy.
This one thing in the chat.
Yeah, there is one question coming through the chat. Let me read it for all of you. It comes from Obika Shunsuke. I'm sorry if I didn't pronounce properly. It says, thanks for your presentation. One question about this PD model. You think that this widespread of alpha-synuclein is caused by the distribution of AV9 or the distribution of alpha-synuclein itself?
Well, we are quite pretty sure that is because of the AV9 spread. So, before getting started with this pilot study, we have performed a number of biodistribution studies using a reporter gene, such as GFP, for example, in order to predict the areas in which we can visualize GFP expression. And I can, since we managed to see that the GFP expression was the predicted one, we finally decided to move into a vector now encoding mutated alpha-synuclein. So in short, yes, in order to answer your question, this can be made possible only because of the retrograde spread of AV9. So there are few other AV capsid variants that has been designed for the specific purpose of retrograde spread. And the most popular one likely is AV2 retro. However, there are some papers showing that there is a huge variability of AV2 retro in terms of performance that is dependent on different species, and it's also it's also circuit specific. For instance, some people have delivered AV2 retro in the putamen of non-human primates, and they realized that it's a circuit-specific spread of AV2 retro. They managed to have a nice transduction of neurons in the cerebral cortex through the corticostriatal projections, but they failed to see any significant level of transduction in the thalamus and in the substantia nigra. And apparently, the same holds true for AV2 retro when being used in mice. So, for studies this type, the selection of the AV capsid, in my view, is absolutely critical. And at least in our hands, the native capsid of AV9 performs much better than capsids such as AV2 retro that has been specifically designed for the retrograde spread of any encoded genetic payload.
All right, thanks, Lancy, and thanks, Obika, for the questions. Any other question?
There is a raised hand by Diego Clemente.
All right, Diego, go ahead.
Hello, good afternoon, everyone. Can you hear me?
Yes.
Yes. Thank you, Lancy, for your question. Oh, sorry for your talk. I have a question about the iFora software. I saw that you have shown pictures with the neurons with a good morphological resolution. So I imagine it for the artificial intelligence is easy to count this kind of neurons. But do you have any experience of? Do you know anyone who has used this software to count something like microglia or astrocytes in an environment full of astrocytic scar with a strong background and all that stuff? Do you think it's useful for that?
It's a very important question and something to be kept in mind. So with iForia, what you need, first of all, is to have a good histological technique. I mean, you need to perform a good immunohistochemistry, because in the end, this is just an object recognition algorithm. So Alphoria will recognize things that can be recognized. So the better you perform the immunohistochemistry with a specific antibody and nice staining, this is going to make Alphoria life much more easy. Another thing is that I don't make mention is that you can count different layers of things. So this is just a single layer algorithm, but you can count, you can prepare a layer, an algorithm recognizing two things together, as long as these are stained in a different way or can be recognized by morphology. So for instance, you prepare an algorithm recognizing neurons versus microglial cells, as long as these two structures are different enough, and this two-layer algorithm can also recognize these structures. So in my experience, the first time I get in touch with Aphoria, because I was totally unaware of this software, was in an APD meeting held in Lisbon in 2017 or 2018, I don't know very well, and I was absolutely impressed. So they have a demo on the booth of Aphoria with a non-human, a human brain section. Is one half of the hemisphere, comprising cortex, thalamus, whatever, stained with Nissen stain. And they have prepared a two-layer algorithm, one layer recognizing neurons stained with Nissen, and the other layer recognizing the nuclei of glial cells. Of course, you cannot recognize astrocytes versus microglial cells, but you can recognize, in an initial stain, the nucleus of a glial cell is quite distinctive for a pyramidal neuron, let's say. And they went through the algorithm just across the entire section as a single region of interest. And in a matter of seconds, you retrieve a message saying, okay, in this section, in terms of the number of nuclei of glial cells, you have, let's say, 100,035 something. So I think that, as long as you can train the algorithm to recognize what you want to recognize, I think you can prepare multi-layer algorithm without any trouble. Indeed, we are doing things here. It also works with fluorescent images. So you can recognize, let's say, you can train the algorithm to recognize red versus green, or red versus or green versus magenta, or whatever. You can create one different layer per different channel, and you can count everything together.
Okay, thank you so much.
Thank you, Diego.
All right, thanks, Diego, and thanks, Lancy. We still have some time for additional questions, if there are any. No more questions?
Okay, fine.
I don't see any raised hand. No questions coming through the chat either.
Okay, it's fine. So thanks a lot for your attention. Thanks a lot for being there. I hope to see all of you in person in the forthcoming CyberNet meeting in Bilbao.
All right, so thank you very much, Dr. Lanciego, aka Lancy. Thanks everyone for attending. Let me just remind you that these webinar series are held every other week, and you can check the schedule for the next few months in the CyberNet website. And so it's time now to close the session, but not before thanking once again to Lancy his willingness to share his work with us. And thank you everyone for attending, and I hope to see you in a couple of weeks in the next webinar.
Yeah.
Okay, have a great day, guys.
Have a great day.
Bye-bye.
Bye.